Hi guys, I am going to help you walk through this Snurfle Meiosis interactive so that you can get all the answers on our Google form or just get a review of Meiosis if you wanted to watch this for some help. So we're going to click start a new game and enter here. And we're specifically talking about this fictional organism that just happens to reproduce sexually. So we know that this is not a real thing, but we're pretending that these snurfles do reproduce sexually and therefore need to do meiosis in order to make the cells that will be used to make babies. So we're going to go through this by clicking Meiosis Interactive. And we're actually going to start with the exact same super long phase that we start with, whether we do meiosis, mitosis, it doesn't matter. Your cells always start with interphase. Interphase is the super long phase of your cell's life, where before meiosis or mitosis, your cell is going to do normal activities, it's going to make proteins, it's going to live its life, it's going to grow, and it's going to copy its DNA. So it is not actually part of meiosis. It's before meiosis, and it does normal activities during this long interphase. So both B and C are correct. So now we're going to zoom in and actually look at what's inside the nucleus during interphase and kind of this whole process. So specifically, these little blue and pink strings are supposed to represent DNA. And when it's uncoiled and stringy and it looks like spaghetti, we call it chromatin. And that chromatin is going to condense into chromosomes ultimately that have a unique number depending on the species. So this snurfle has one DNA molecule from the dad, the blue one from the mom, the red one, pink one, whatever. And we're going to just look at this cell with only 2 pieces of chromatin. Now keep in mind, if you're a human, you're going to have 46 chromosomes. So we're really simplifying here just to make it easy to look at. So this DNA has certain genes on it. We talked about genes as basically parts of your DNA that code for traits. So we could use big letters to represent dominant genes. We could represent recessive traits with little letters. That's why they have a big G there for the yellow fur and the little G for the green fur. Keep in mind, if you switch over to your Google form, if you are filling this out, does ask you what color the big G is for, which again, if you paused here, the big G is for yellow fur. So before meiosis can occur during that interphase, we're going to have our DNA copy itself. So you click on the DNA, we're going to make sure we copied our chromatin. So DNA does replicate during interphase. Chromatin is just DNA, and genes are just segments of DNA. So all of these are true. So we're going to make sure we pick that all of these are true. After interphase, meiosis will begin. Meiosis is basically a process that we're going to utilize to make sperm and eggs, which we call gametes. So why do we do meiosis? To make sex cells in order to reproduce ultimately. So meiosis is made of many steps, just like mitosis, prophase, metaphase, anaphase, telophase. Meiosis is going to be the exact same group of steps, but we're going to divide them into 2 separate divisions, meiosis 1 and meiosis 2. So the PMAT, pretty miscall, always teaches prophase, metaphase, anaphase, telophase is the exact same order for meiosis. We just do everything twice. So it's prophase 1, metaphase 1, anaphase 1, telophase 1, and then we do it all again in meiosis 2. So I asked you again, what's the same order of phases for meiosis 1, meiosis 2, and mitosis? We always go through PMAT. PMAT, pretty miscall, always teaches prophase, metaphase, anaphase, and telophase. So now let's actually look at them. In prophase 1, you're going to see the same kind of things that you would see in mitosis, but plus a couple extra things. For example, when we are doing prophase, we know that we have the chromosomes condense and become visible. So all that DNA is going to coil up and we're going to actually see chromosomes for the first time, which is important so that we could very easily move my chromosomes around when I need to. So these chromosomes are going to do something special in meiosis that they don't do in mitosis. In meiosis, when we have chromosomes that you got one from your dad and one from your mom that are the same size, they're the same shape, they have the same genes, we call them, look at this big H word, homologous. And homologous chromosomes are important right here. Homologous chromosomes, because they do something special in meiosis. Each half of that chromosome is still called a sister chromatid, and those 2 pieces are identical. But the sister chromatids of the 2 homologous chromosomes are going to ultimately do something unique here. So again, in prophase, we want to get rid of that nucleus, that nuclear membrane, because it's in the way. It's like a cage. So we're going to click on the nucleus to make it go away in prophase, right? Then we're going to have the pair of homologous chromosomes get together and get cuddly and snuggly. And this is called a tetrad because there's like 4 chromatids. That's why they call it that. So I'm not going to ask you about the word tetrad on your test, but just in case you see that, you know that in prophase, sorry, we're still here, we are having those chromosomes pair up. So the homologous chromosomes get next to each other, they get snuggly, they get cuddly, and in fact, they could get so cuddly and snuggly and close together that they actually swap pieces with each other. So what did happen in prophase 1? The chromatin did condense to form chromosomes. The nucleus went away. The homologous chromosomes paired up, but they did not line up down the middle. So 1, 2, and 3 would be my answers, which is what you're going to select on here like that. And then in metaphase, guess what? The exact same thing in metaphase is going to happen in metaphase of meiosis. So again, this slide just says, hey, don't forget something weird might happen here called crossing over where they swap pieces, but there's a separate interactive about that we just aren't doing right here. So when we do metaphase 1, here's something that's a little bit unique. Yes, the chromosomes line up in the middle, right? Metaphase middle, but they no longer line up in a single file line. They line up next to each other in a pair. So they are literally side by side. That's important. So tetrads line up down the middle of the cell. The chromatin does not condense. That was prophase. Homologous chromosomes don't separate. That's anaphase. So it's just A that they line up down the middle. So in metaphase 1, the tetrads line up down the middle of the cell. Look at the next step, which is pretty misculp always, anaphase 1. In anaphase 1 of meiosis 1, the chromosomes are going to split. But instead of splitting individual sister chromatids, we're going to pull an entire chromosome from a whole chromosome. So now those homologous chromosomes are being separated from each other. So during anaphase 1 of meiosis 1, we have our homologous chromosomes getting separated and moved to opposite ends, which is C, which is what you're going to pick right here. In telophase 1, telophase 2, new nuclear membranes. So we're going to make each of these chromosomes basically get back into a nucleus that we disintegrated back in prophase. So you can start to see my cells beginning to split. You can see it pinching in. The nucleus is going to reappear. We are ready. Now it's time for that last step of the cell cycle called cytokinesis, like telekinesis, but cytoplasm. So we're moving the cytoplasm. So we're going to split the cell in 2, and we're going to do that right here by dragging this, and that's cytokinesis. Remember, this is an animal cell, so that's why it's easy to pinch in. Guys, now that I've split half of the chromosomes into one cell and half of the chromosomes into the other cell, we no longer call these cells diploid. Diploid implies that there's 2 of each chromosome. A haploid cell has half the normal chromosome number. So they're already haploid. They're already halved. If this was a human being, they would have half the normal chromosome number. So during telophase, the nucleus reformed. Each new cell that's formed after cytokinesis is now called haploid. So if these were humans, 23 would be in here and 23 would be in here. So why are we doing meiosis 2? If I already halved my chromosome number and I want sperm and eggs to have 23 in each, why are we doing this again? Because look at those chromosomes, right? The chromosomes are still intact. They're still X's, and we need to fix that. So meiosis 1 looks just like mitosis. We're going to have cells at the beginning that are diploid, just like in mitosis. They have 2 of every chromosome. There's 46 if it was a human. By the end of meiosis 1, however, we now have 2 haploid cells, which is different than mitosis. So now let's look at those cells and start all over and do prophase 2. In prophase 2, just like in prophase 1, the chromosomes are going to be visible and the nucleus is going to go away. But goodbye, nuclear membrane. Then we're going to see something different. The tetrads do not form in prophase 2 because there's no pairing up. There's no homologous chromosomes to get together. The nucleus does not go away. No, that's not true. There are no chromosomes. No, that's not true. So only A is true about prophase 2. And prophase 2, the tetrads do not form, right? We don't have pairs, okay? In metaphase 2, they still line up down the middle, but now it looks like mitosis. They line up in a single file line down the middle of the cell. That's metaphase 2. In metaphase 2, the chromosomes are going to line up down a single file line down the cell. And I did include some pictures in here to help you know where you're at if you're doing my Google form. In anaphase 2, apart, my chromosomes are going to get pulled apart, and now just like in mitosis, each sister chromatid is getting pulled, not a whole chromosome and a whole chromosome. That was anaphase 1 of meiosis 1. So now in anaphase 2, the sister chromatids separate and move to opposite ends of the different poles. Now I'm at the end again. Telophase 2, I'm going to have 2 new nuclear membranes form in each of my cells, the chromosomes are going to decondense and go back to spaghetti ultimately here. And I'm going to do cytokinesis and split my cell again. And this time each cell will be split, so that's going to make 4. What happens during telophase 2? We get a nucleus, the chromosomes go back to chromatin, 4 haploid gametes are being formed. All of those are so true. What are the final results after we're done? Look right here. When we split our 2 cells into 2, I'm going to end up with 4 cells that are all haploid. This has 23 chromosomes. This is 23. This is 23. This is 23. If this was a human, right? They each have half the normal number. So that's why they're called 4 haploid gametes. So now that we did that, we have 4 haploid gametes as our end product. That's different than mitosis, where you had 2 diploid cells, like you had 2 cells that had the normal number of chromosomes in them. So this is our overall look of meiosis. You start with a cell that's diploid. It has all the chromosomes, 2 of each. It has homologous pairs. By the end of meiosis 1, you have 2 cells that each only have half the normal number. But look at those chromosomes. They're still X's, and that doesn't work for us. So we have to do meiosis 2, and we end with 4 haploid gametes. And remember that gametes are eggs and sperm. So we're going to pretend that these are eggs that we just made for the girl Snurfle. Okay? So we're going to do this again with the guy Snurfle, but remember that it's the same process in the sense that we're just doing meiosis 2 times. So we're just doing it a little quicker so you can kind of review. It says, okay, let's make these sperm. We're going to make prophase happen, which means nucleus disappears. The chromosomes are visible. We're going to have the homologous pairs get cuddly, cozy, maybe do some crossing over. In metaphase 1, the chromosomes line up in pairs down the middle. In anaphase 1, the chromosomes split apart in the whole chromosome, not each sister chromatid. And to telophase 1, 2 new nuclei form around my chromosomes. And I'm going to do cytokinesis probably around the same time and split my cell in 2. Now I have 2 haploid cells, but I'm not done because look at those chromosomes. They're still intact. I'm going to have to do meiosis 2. So we just finished meiosis 1. Now we go to meiosis 2 and do the exact same 4 steps. In prophase 2, the nucleus is going to go away. We don't have any homologous pairs. We don't have any crossing over because it already happened in meiosis 1. The chromosomes, are going to line up down the middle, in metaphase 2, but they don't line up in pairs, single file, honey. In anaphase 2, the sister chromatids get pulled apart. So now they're individual chromosomes. And then in telophase 2, we're going to have 2 new nuclei form in each cell, so that when I'm done here and I'm going to have the chromosomes go back and uncoil, I can do cytokinesis and split and have 4 haploid gametes. So we just made the sperm for the dude. Okay, so we did it twice, and you're done. So that is going to take you to the end where it says, what do you have to do after you make the eggs for the girl? You have to make sperm for the boy. And then I asked you a question. How many chromosomes are in this picture? Just to review. So if you look at this picture, there are 4 chromosomes in that picture. 8 chromatids, right? Because a chromatid is half of a chromosome. That's it. You're done. You saw meiosis. And remember that the product of meiosis is 4 haploid gametes, sex cells.